Me?
Yes, yes. Just check.
Okay.
Oh, that then. Good morning, everyone. I welcome you all in the third B section of the sixth SIG conference on
on
microbial
microbial
processes and
processes and
products.
products.
For this
For this
session,
session,
we have
we
Professor
have
Paramjit Panesar and Professor Samir Khanal as the moderators of the session. And we have six speakers in this: Professor Hector Ruiz, Dr. Palanivel Satish Kumar, Dr. P. H. Lee, Dr. S. Venkat Mohan, Dr. R. Parthasarthi, and Dr. Amiten. I welcome you all to the session. Now I'll introduce our session moderators. I'll introduce Professor Paramjit Panesar. Professor Paramjit Panesar is currently working as Professor and Dean at Sant Longowal Institute of Engineering and Technology, Punjab. His research focuses on the value addition of agro-industry byproducts, prebiotics, food enzymes, bioactives, etc. He has published over 150 scientific papers, 35 chapters, and 9 books. Professor Panesar was awarded the Boycast Fellowship by DST Government of India. In recognition of his work, he was selected as Fellow 2018 by BRSI, Fellow 2019 by the National Academy of Dairy Sciences India, and prestigious INSA Teachers Award 2020 by Indian National Science Academy, INSA. Professor Panesar has also been listed for 2020 and 2021 in the list of world ranking of top 2% scientists published by Stanford University, USA. Now I'll introduce Professor Khanal. Professor Samir Khanal is Professor of Environmental Engineering, Department of Molecular Biosciences and Bioengineering, University of Hawaii at Manoa. He received BS in Civil Engineering from Malviya National Institute of Technology, Jaipur, India, and MS in Environmental Engineering from Asian Institute of Technology, Bangkok, Thailand. He obtained PhD in Environmental Engineering from the Hong Kong University of Science and Technology, Hong Kong. Professor Khanal is a globally recognized researcher in the field of anaerobic digestion, nanobubble technology, aquaponics, and waste to resources. Professor Khanal received highly prestigious Board of Regents Medal of Excellence in Research 2018 and CTHAR Dean's Award for Excellence in Research 2016 UHM and Elsewhere's Impactful Research Award 2018. Next is the Pandey Research Excellence Award 2021 and Outstanding Alumni Award HKUST 2021. He is an associate editor of Bioresource Technology. He has served on various committees of other professional societies, including International Bioprocessing Association. So, with this introduction, I hand over the floor to the session moderators, Professor Paramjit S. Panesar and Professor Samir Khanal. Thank you, everyone.
Thank you, Dr. Sunita Barjani, for the introduction. I, along with my fellow moderator, Professor Samir Khanal, welcome you all to this session. The session is focused on the microbial processes and products. We all know microbes have played a very important role in management of the environment as well as the treatment of industrial byproducts. So we have the six speakers in the session, and we have two speakers in the physical mode and four speakers they are online delivering their lecture. So I will just briefly introduce our panelists for the today's session. Uh, First panelist is Professor Hector Rios. He is from University of Colia, Mexico. Dr. Hector obtained his PhD in Chemical and Biological Engineering from the Center of Biological Engineering at University of Minot. Dr. Rios is a full professor and founder of Biorefinery Group at Autonomous University of Coahuila in Mexico and the leader of biomass pretreatment stays in the cluster of bioalcohols in the Mexican Center of Innovation in Bioenergy in Mexico. Dr. Rios is a editor-in-chief of Bioenergy Research Journal, associate editor of biotechnology for biofuels, and editorial member of Industrial Crops and Products and Biofuel Research Journal. So he has published various papers in high impact journals, and his research work is focused on particularly biorefining and the production of high value products, biofuels, biomass fractionation using hydrothermal processing, and bioreactor design. Our second panelist, Professor Plainwall Satish Kumar, he is from South China Normal University, China. Dr. Satish Kumar is presently working as associate professor in the School of Chemistry, South China Normal University, China. Dr. Satish has received his doctorate degree from Prihar University, India. After a PhD, he started his research career as postdoctoral fellow in the Yonwang National University, South Korea. He has different high impact articles to his credit, and he is also serving as a managing guest editor in some of the special issues in several reputed journals. Our third panelist is Dr. Lee, Dr. P. H. Lee, and he is from the Imperial College, London, United Kingdom. Dr. Lee obtained his PhD in environmental science from Iowa State University, USA, and he has joined as a senior lecturer in Imperial College London in 2019 after positions at Hong Kong Polytechnic University, China. His current research interest, they are interactions between the microbiomes and conductive materials, microbial responses to sub-nano levels of oxygen and antibiotics, quantum machine learning on full-scale engineering databases, etc. Our fourth panelist, Dr. S. Venkatamohan, he is from Indian Institute of Chemical Technology, Hyderabad. Dr. S. Venkatamohan has been working as senior principal scientist in IICT Hyderabad since 1998. Dr. Mohan was a visiting professor at Kohato University, Alexander Humboldt Fellow at Technical University of Munich, Germany, and Kyung Hee International Fellow, South Korea. Dr. Mohan is placed in second position in India and 29th position in world in the field of biotechnology by Stanford University. Studies on 2% top level scientists. He has guided 29 PhDs, 100 MTechs and BTech students, and he is the recipient of most coveted Santi Chavru Bhatnagar, a prize for the year 2014 in engineering sciences from Government of India. Our fifth speaker, he is Dr. R. Parthasarthi. He serves as a principal scientist in Indian Institute of Technological Research, Lucknow, and computational technological facility, and leading VIRAC, BioNest, and DSIR IITR, Environmental Monitoring and Interventions Hub. His work is mainly focused on the non-toxic solvent development for bioprocessing and even developing the low-cost technologies for the industry's environmental scenario. He has received several awards, including Director's Fellowship, uh, Lewis Almus National University, Fellow of Royal Society of Chemistry, Dr. APJ Abdul Kalam Award, Professor S.B. Chitolkar Memorial Award of VRSI, and Young Associate of Indian Academy of Sciences, 2008. Sixth panelist is Dr. Amy Tan. He is from Department of Civil Engineering, the University of Hong Kong. Dr. Amy is assistant professor at the Department of Civil Engineering, University of Hong Kong. She holds a PhD degree in Civil and Environmental Engineering and a bachelor degree in biological sciences from Singapore. Dr. Tan research is focused on waste to energy, waste to biosource, then biotechnological innovations in sustainable development from the organic waste. And she has also received several awards, including Environmental Paper Award from Hong Kong Institution of Engineer 2019 and Outstanding Paper Award for Young Engineers Researchers in 2016. So I welcome all the panelists to this session. All the panelists, they have high research impact in different areas. They are master of their domain. With this, I first invite Dr. S. Venkatamohan to deliver his talk on the biorefinery tool for sustainable bioeconomy. Dr. Venkatamohan, please.
And greetings to all the panelists as well as delegates. Very good morning. Today I would like to present the biorefinery, a tool to suspend circular economy. First, I would like to thank the International Society for Energy Economy and Sustainability. The audience questions. Data which pertain to media exam. Present the media consumption model, particularly looking into causes and global material consumption is 4 times since 1970, whereas population is 3 times. According to estimate the records, the domestic material consumption in 2017 is about 100 billion tons per year, accounting for 11 tons per person per year on average, which by 2050 will increase to nearly 200 billion tons per year. And this is a huge range. And apart from that, the waste generation, particularly from 2015 to 2017, doubled. In 2017, it's about 32 billion tons. It's a huge by 2050, the population is 25%, where India accounts for about 80% of the population. If you continue to do like this and produce like today, by 2050, we will need 3 times more resources, by 50% more fuel, 70% of more food, feed and fiber, as well as 50% more of water. And adding to this, about 70% of people by 2050 is near the national. So this will again add a lot of stress, even though 2% of global area is being covered by urban markets, but 80%, close to 80% GDP, but also consume 75% of natural resources by producing 50% of global water. It's a significant thing needs to be considered, particularly in the future, with respect to materials as well as urbanization. So something we have to move towards responsible consumption. And they in one of the strategies towards moving from linear to circular economy is one of the sustainable aspects. But as per present, 8 to 9% according to 2020 figures, resource recovery is happening, and significantly 92% is going for landfills or some of the waste things. So it's a very critical requirement in order to move towards something that sustainable. And circular economy model is one of the vital model as I look into, because it has all kinds of sustainable interventions wherein the resource recovery is the major aspect, and the resources should be used in a sector mode by symbiotic approach without wasting the material. As everybody knows that this is based on three principles, and I am more interested in biodiesel and in biological process. And about 25% of the resources which is being used is from biological source. Using biological process as well as biological material, that is waste material, by going towards bioeconomy is one of the sustainable strategies. So for going to circular economy, by using waste as a feedstock, we find waste in a significant amount. And in Indian context, we have a huge amount of waste in the form of municipal surface, as well as domestic sewage, apart from fecal sludge and industrial wastewater. And there is no depth of material like causes. Every year there is 10%, particularly in municipal surface. However, we are only using 4% of resource recovery, particularly towards energy. So to go towards something refinery aspects based by chemical lifestyle, we require sustainable technologies, particularly bioprocessing technologies. And most of the process which is at present we are in high level when we think about transition by taking waste. And some of the technologies are the PRL4 and distributed widely. But it can be any kind of domestic waste or industrial waste or including CO2 that can go with biological processing to produce products that have significant applications apart from waste generation. So in this direction, I would like to say that we are working on four different domains, four costs, I would like to say broadly, and green hydrogen by taking the four dark fermentation. The process has advantage the convention process but last step has to be one mole of glucose. We can produce about 4 moles of hydrogen, which is a very important green hydrogen route, I would like to say. And 1 kg of CO2 is 400 tons. So we are able to set up a pilot plant with type of ministry of and huge capacity. This is the photographs. We are trying to upscale it by these by using about 1 lakh ton of hydrogen. To add to carbon nitrogen, there is another advantage which platform companies all the hydrogen when we go in cascade as one mode of citric acid, so we are also focusing on generating enhancing fatty acids towards addressing the golden fatty acid platform with respect to this and increasing the fatty acid production from these sources. So it is a win-win situation. Apart from that, second first year working on is microbial processing. Huge advantage initially starting with microbial. But when we look into its application during its 15 years of time, and broadly we can call it as microbial electrochemical process as diversified into different areas, particularly with respect to biorefinery, bioelectrochemical systems and microbial electrolysis are very important applications in microbial chemistry that has huge applications, huge emerging the common organ including biomass and bioplastic. So, in order to address the refinery, we try to take estrogenesis at the first phase and factors to be produced and outlet from hydrogen reactor and integrate it with any of the biological process to produce second product by maximizing the resource recovery. We can integrate with the anaerobic process by methane production or with microbial electrolysis for additional hydrogen, or we can integrate with the anaerobic process to produce bioplastics in the form of polyfibrous sulfate, or can go for organic farming, or with isotrophic organic calculation, we can convert matter into biodiesels, or with microbial chemistry, we can produce electricity. By way of this integration, we are able to recover resources to 90% apart from wastewater treatment, either in the form of two energy forms or one energy, one food. It's a model which we have developed in ISBT, platform waste production model, and we can different products in integration based on wastewater, usually go to microbial person, then for explosion, the fourth process, you can go to any of this process to convert different forms. They're not going to do it because of lack of funds. It's the OD of the biofriendly facility in ICT. They are also working in different kinds of industries like sugarcane industry, where we can use the waste, the sugarcane bagasse as well as spent costs, which are important in feedstock, to convert into different kinds of chemicals that will have less importance, and also trying to use self-sustainable photovoltaic process. It goes into approach by taking the and feeds to convert it to biomass and then to subsequent chemicals in a closed loop approach without giving any input. Likewise, these kinds of process will have advantage and also support majority of the sustainable development goals. However, it required very multidisciplinary approach research as well as very concerted policy in order to make it into available to the existing market. Thank you very much for your kind attention.
Thank you, Dr. We have the next speaker, Dr. Hector Arius. He's from the University of Mexico. He will be delivering the lecture on high solid loading processing of biomass. Professor Hector, please.
Yeah, thank you. Well, okay, I can see my presentation. Well, first I want to thanks for the invitation, Professor Pandey, Professor Sunita, and Professor Panacer for this conference. Well, next, please. Next. Well, okay, I am in Mexico, in the north of Mexico, 300 kilometers to Texas. It's the state of Coahuila, and I am working in the Autonomous University of Coahuila, and in specific in the biorefinery group. This is a group for focus in terms of the valorization and fractionation of biomass using different kind of raw materials in terms of the biorefinery concept and in terms of the circular bioeconomy for produce high value compounds and biofuels, in the specific, the bioethanol. Well, okay, the next, please. Well, in terms of the processing in these days for produce these high value compounds or biofuels, it's important developing different kind of strategies. For example, it's an important to developing different process using high-hat solid loading. What is the reason with this situation? At the end of the process, we need to obtain high concentration of sugars, high concentration of these compounds in terms of reduce the cost. And in specific, in our research group here in Mexico, we are trying to develop this kind of process using hydrothermal pretreatment as a principal process for the fractionation of biomass. This is very important because we can obtain that solid phase and liquid phase reaching sugars and obtain after of this stage different kind of phases for try to convert in high value compounds. So it's a very important to development or try to find these strategies in the batch mode operation or in continuous operation using high-hat solid loading. So next, please. So what is the situation, or what is the bottleneck in terms of the developments for second generation or third generation biorefineries? In this case, of course, is in terms of the techno-economical situation or issues that we have in the process. In this case, for example, the feedstock, the enzymes used, or the pretreatment, in this case, the fractionation processing, are the most contributions stage in terms of the second generation. So we need to focus in this kind of a stage for try to reduce the cost, for try to optimize the process in this case. So in our group, we are more focused in terms of developing the hydrothermal pretreatment for use different kind of phases. Next, please. So in this case, the hydrothermal processing is considering an environmental friendly process that we can use only water for try to hydrolysate, for try to modify an extraction of different compounds for lignocellulosic biomass using high temperatures and high pressures and using only water. These kind of processes are very selective because we can obtain in the liquid phase, for example, you can see in this photo, the liquid phase is an hemicellulose fraction rich in 5 sugars, and the liquid phase, and you can see in the solid phase, the lignin and cellulose, in this case glucan, we can convert in the next stage for the enzymatic hydrolysis. So it's very important to try to development this kind of process for the next stage in terms of the enzymatic hydrolysis. So what is the other advantage, for example, for the hydrothermal pretreatment? At this moment, for the pilot scales, for the industrial scales, we can develop it using different kind of strategies. For this use, for the batch mode or for the continuous mode operation. So in our opinion, this is considering an environmental and a good and a promising process for the biomass fractionation. Next, please. So other important things in terms of the biorefinery is try to not only produce this kind of biofuels using high-hat solid loading. We need to produce other high-hat value compounds. For example, this is the schematic of the general process using hydrothermal pretreatment or fractionation of biomass for try to produce the oligomers from hemicellulose fraction. Using these oligomers and biofuels, we are close this concept of the biorefinery, and of course we can decrease in general terms the cost for the second generation development biomass. The next, please. So, and the other important things for this panel or this session in the microbial and the bioproducts is try to using this high solid loading. As I mentioned, the pretreatments and the enzymes are the most contribution in terms of the development second generation. So it's very important to try to design bioreactors using this high solid loading, about 15 to 30% of biomass for try to produce high concentration of sugars. So it's important to put attention in terms of the designs of bioreactors and use this solid loading. The next, please. So in our case, in general terms, we are applying the solid loading for agave bagasse here in Mexico. The agave bagasse is a promising raw material. It's a subproduct for the alcoholic beverage. The name is a tequila, very known here in Mexico. And we use this residue, agave bagasse, using hydrothermal pretreatment for try to produce a rich solid phase in cellulose for convert in fermentable sugars. As you can see in this graphic, we can typical behavior in terms of the degradation compounds, furans in this case, and the glucose and hylose in the liquid phase. But in the solid phase, we can increase the concentration of cellulose, and it's a very good point in terms of conversion for the next stage for the enzymatic hydrolysis. So using hydrothermal pretreatment is a good process for trying to optimize the process in the increase and the concentration of cellulose in the solid phase. Next, please. So in this case, we can see in these photos the modification of the structural in terms of the cellulose concentration using these kind of conditions. And the most important is try to design these kind of reactors and use 25% solid loading, or try to produce more or less 200 grams per liter of glucose. This is a good concentration in terms of the next stage for produce high value compounds or biofuels, in this case, for example, ethanol. We can convert in 100%, we can produce 100 grams per liters of ethanol. It's a good concentration. So this is at the next stage to try to find this process for using high solid loading. It's a good point for the next stage in the development of second generation. Next, please. And in terms of the conclusions for this short presentation, it's only for try to see the importance of use and try to develop in this process using high solid loading. Is very important to try to reduce the cost, to try to increase the yields, to try to increase the concentration of the fermentable sugars for produce other compounds or biofuels. It's a very important, and I think that at this moment the bioprocessing we can focus for trying to develop it. The next, please. And okay, this is the team here in Mexico. This is my email for the we have touch. And the next, please. The last slide. And to invite you to send to the last word to the bioenergy research for different kind of topics. Thank you so much for your attention.
Thank you, Professor Hector. Now, we will handle the next session, the next lecture session. Professor, please.
Hello, good afternoon, good morning, good evening. Now, our next speaker is Microbial Satish Kumar from South China Normal University, Guangzhou, China. And he will be giving a talk on modified microorganisms and their product uses in agriculture and environmental applications, current updates. Satish Kumar, now you can go ahead.
Yes, sir.
Dr. Satish Kumar, please, to start the lecture.
Good morning, once again, good morning to all. First, I'm Dr. Microbial Satish Kumar from South China Normal University. First, I would like to thank our chairman, organizing members, moderators, panelists, and all conference joiners for the International Conference Sustainable Energy and Environment Challenges challenges. So I would like to share some information on the current updates on the modified microorganism and their potent products usage in agricultural and environmental applications. Can I share my slide? Is it possible? Discussing about several microorganisms have been used to solve different agricultural and environmental issues Agriculture is just the crop production, plant health production, soil health management. Agriculture is just the bioremediation of organic, inorganic pollutants in soil and water. Apart from this, yeah, the microbial process has been used in various processes such as the wastewater treatment, recycling of agricultural industrial waste also. However, a natural and wild microorganisms are very less potential for the real applications. Developed some of the technology, that is the genetically engineered microbial strains for the real applications to manage these issues. So mainly four tools have been used in this genetically engineering modified organisms. First one is the gene transfer method. This method is mainly used to deliver the selected target gene into the desirable host. Second one is the cloning vector. This cloning vector is mainly used to clone and increase the copy number in the organisms. Third one is the promoter. This one is mainly used to control the expression of target genes. The last one, selectable marker genes. This markable gene is mainly used to identify recombinant microorganism, which after the cloning process. Next. Next slide, please. How to change next stage. Okay. So first, before entering into the topic, we should know how we can make the genetically engineered modified organisms. So there are mainly 11 steps for the genetic engineering microorganism production. First one is the selection of target organisms. Second one is the production and isolation of DNA fragments from the target organisms. Third one, from the DNA fragment, we will isolate the gene of the target. Then the fourth one is the selection of desirable vectors. Fifth one, cut off the suitable restriction sites by the with the help of restriction enzymes for the introduction of the gene into the vector. Sixth one is the insertion of target gene into the vector. After that, the seventh one is the vector cloned with the desirable gene. Eighth one is the selection of the host cell for the transformation of the vector that contains desirable gene of interest. So after the vector construction, the constructed vector will be introduced into the host cell. That is the ninth step. Then the 10th one, the reproduction of host cell produce multiple copies of cloned genes within vector. This is the mechanism and process of genetic engineering microorganism productions. Next one. Next one. So still now there are so many modified organisms have been produced by various research groups. These are the first microorganisms: bacteria, fungi, and algae. If you're seeing about the bacteria, mainly Pseudomonas fluorescens, MK44, Pseudomonas putide, E. coli strains, Agromobacter, and Agromobacter. Agromobacter, Sponginium indigum, Sponginium japonicum, Pseudomonas species BF13, Bacillus subtilis 168, YCM, SM, and also some of the algal species, the following algal species. And if you're seeing about the fungi, mainly the modified organisms are Schizosorium, Xanthium, Florus Vostratis, Magnus Paraxidis 2, and then the recombinant Heterogate Rhizosphere, Saccharomyces cerevisiae, ML201, Saccharomyces cerevisiae, ML1A, ML201, especially disease. X, Y, one, Arabillus, Nurlens, Gagpa, Aspergillus, Boracia, LB, H, H7. These are the main genetically engineered microorganisms have been practically in several applications, including food, agriculture, and environmental applications. Next slide, please. So next slide, please. Okay. So if you are seeing about the application of agricultural application of genetically engineered microorganisms, mainly it is used for the production, enhancement, and production of agricultural products. So for in this one, biofertilizer has been used as the agricultural products production, and also as well as the biopesticide for the agricultural product production. So, for example, biofertilizers, nitrogen-fixing genes, Mifel and Ca has been used in soil fertility enhancement. So, this gene inserted Rhizobium mellitus bacterium have been used to induce the nitrogen fixation, nitrogen fixation process. If you see about the bioprocess, biopesticides, the toxic gene of the basis turigenesis is inserted. Species have been used to control the pests. For example, to control the insects. This pesticide is highly toxic to the insect larvae. So these are the main application in the agricultural part. It is mainly used as the fertilizer and biopesticides. Next slide, please. Next slide, please. Okay. So, if you're saying about the environmental applications, the first the superbug was developed by Ananta Mohan Chakrabarty in 1975. He received a patent on a genetically modified Pseudomonas putidei in 1980. He was the first person to win patent on a living organism. So after that, several genetically modified developed for the environmental and agricultural applications. So if you can major application in the environmental or the heavy metal removal detoxification, degradation diet, industries effluents, degradation of organic xenobiotics such as the pharmaceutical active compounds, personal care products, pesticides, etc. So apart from this kind of genetically modified organisms have been used in the oil pollutant degradation also. So we can discuss in detail in the next slide. Next slide, please. Okay. This table shows the list of modified organisms used for the degradation of heavy metal ions. So nearly around 10 organisms have been reported for such kind of heavy metals ion the modification process in the genetic engineering. For the degradation of mercury, Deinococcus radians have been used after the modification with the MEC-H transporter gene. Similarly, the MEC-C, that is the mercuric ion transporter gene, incorporated acidothiobasilus peroxidins have been used for the mercuric degradation. Similarly, nickel transportation system products such as the NIX-A gene incorporated E. coli SE5 strains have been used for the nickel degradation from the aqueous solutions. The sodium species constructed with the Hg transporter system and the organomercuric layer used for the mercuric degradation. And also enhanced green fluorescent protein, that is the EGPA, and the methylparathione gene incorporated Pseudomonas putidei X3 is reported for the degradation of methylparathione and cadmium. These are the most important modified organisms used for the heavy metal ions. However, most of these organisms used in the laboratory scale, but not in the real scale. For real scale experiment, it is essential for this field applications. Next slide, please. So this table shows that the modified organism used for the dye degradation. Pseudomonas fluorescens stains, baseless amyloliquifications, D501G variants, Streptococcus lividens stains, baseless, modified with the co-TL lacase mutants is used for the degradation of different dye compounds. So if you're seeing about the Pseudomonas fluorescens modification, SZ6 and SD is that three gene incorporated Pseudomonas stain used for the degradation of different kind of dye compounds. Similarly, site-directed mutagenic baseless amyloid liquefaction D501G variant is used for the indigo dye, and baseless evidence after the homo expression is used for the degradation of indigo dye. Baseless is mainly used for the degradation of everything. This kind of organism have high thermostability for the degradation process after modification. So next slide, please. Next slide, please. Okay. So this table shows that the organism is used for the dye degradation. Sorry. Next. This table shows that the degradation of different xenobiotic compounds. So till date, mainly Pseudomonas organisms have been used for the degradation of organic after modification. Pseudomonas dimutant after the recombinant expression have been used for the organophosphorus compound degradation. The TMFS gene expressed alkylagens organisms have been used for the degradation of variety of organic and organic compounds degradation. Pandoria organism after modification with lacase gene expression have been used for the degradation of xenobiotic various xenobiotic compounds. This process have been achieved by various primary metabolic pathway process. That the SDS degrading gene expressed Pseudomonas is also used for the degradation of pentafluorosulfide subunit aminophenyl compounds. Apart from this, alkaline species also have been reported for the degradation of 2,4-D after the modification with NAH or NAHU gene expression. So these are the bacterial stains have been used for the reported for the organic xenobiotic compound degradation. And next slide, please. So this table shows that the biodiversity of pesticides. So pesticide, if you are seeing about the pesticides, the modified organisms such as the E. coli, Moraxella, Spongium, Pseudomonas putide, Tutti KT244, and Spongium abamilius, Pseudomonas putide have been reported for the degradation of variety of pesticide. Example, organophosphorus pesticides, phenyl nitrophenyl compounds, organophosphorus pesticide, degradation of methyl parathene and carbohydrate. So if you're seeing about the E. coli, often modified with PLBS red and PLOPH and PDS plasmid insertion have been used for the degradation of organophosphorus compound degradation. Similarly, sulfur express organohydrolase enzyme expressed Moraxella have been used for the phenyl nitrophenolic compounds and organophosphorus compounds. Methyl parathene hydrolase gene incorporated spongial organisms have been used for the methyl parathene and carbofuran organism. And Pseudomonas putide incorporated with promoters J23199 and PH, that is the pyrethylene hydrolysing carbolase enzyme, and the MPD and OP degrading gene, the incorporated stain used for the degradation of ferronitrothene, chlorophylls, methyl parathene, cypromethane, such kind of pesticides. So, and also, if you're seeing about the Pseudomonas putide X3 modified enhanced gene, fluorescence protein gene, and methyl parathene have been used for the different kind of soil microsome microorganism with cadmium and as well as the methyl parathene pesticides. So these are the bacterial stains have been used for the degradation of pesticides. Next slide, please. So this table shows that the list of modified organisms used for the degradation of organic pollutants. If you see about the Pseudomonas putrida, this is the most important organism used in the environment for the pollutant remediation. This organism used to detoxify the toluidine benzoate compound also. Common monas testogens is to the monochlorobiphenols organism. Pseudomonas pseudoalkalogens also used to degrade trichloroethyl, trichloroethyl, toluene, benzene, and fluorobiphenols also. Pseudomonas putridae, Pseudomonas tetraidis used to degrade cyanide phenol compound organisms. In the Pseudomonas putrida, different stains use all the naphthalene, trichloroethylene degradation also. So these are the list of organisms used to degrade pollutants. So next slide, please. Okay, this table shows that the mechanism of oil degradation by genetically modified organism. This genetically modified organism in the biological process, it will produce oil degrading enzyme oxygenase under aerobic respiration process. So these enzymes mainly attack the hydrocarbons and produce carbon dioxide and water molecules. This is the mechanism of oil degrading process by genetically engineered organisms. Next slide, please. Okay. This table shows that the genetically modified organism used to remediate oil pollutions. So these are the bacterial stains have been used reported for the oil pollutant remediation. Pseudomonas putridae, Pseudomonas fluorescens, Rhodococcus, Pseudomonas aerogenes also have been reported after modified for the oil degradation. For example, NaH and XOIL plasmid hybrid with plasmid CM and octa used for the degradation of crude oil samples, and so Pseudomonas fluorescens used for the naphthalene. Then the Pseudomonas R is the single pathway in the degradation crude oil also. So these are the most important organisms reported for the oil pollutant. It is mainly for decontaminate the polluted
बंद करेंगे ना मेरे को थोड़ा टाइम चाहिए.
So next slide, please.
प्रोजेक्ट जैसे बंद करेंगे 15 मिनट 10 से 15 मिनट बाद ही ऑन कर सकता हूँ. कुछ हाई लेंथ होता है एकदम करें तो फूकने के टाइम.
So this table shows the degradation of radioactive compounds using the chemically modified organism.
अरे मैं आपकी बात समझ रहा हूँ. स्टेशन जब चल रहा है तो 2 मिनट रोक के सकते हैं. लाइव है ना वो. मैं ये कह रहा हूँ, आप श्योर करो कि या तो पावर में कोई इशू नहीं है प्रोजेक्टर में.
Similarly, Rhinococcus thermo.
किस रूम में कराना है, क्योंकि उसमें तो फंक्शन की तैयारी होने लगी.
एमराल्ड 1 में.
हाँ, एमराल्ड 1 या 2 में किसी अपनी तैयारी चल रही फंक्शन.
सर को बोल के 2 में करवा लेते हैं.
2 में ही होना है.
2 में होना है.
कल बात करी थी, मैंने पूछा था. हाँ, तो उधर वाले 2 में ही होना.
प्रॉब्लम वही है ना, उनके पास वहाँ पे बैकड्रॉप नहीं है. और दूसरी चीज़ ये है कि अगर आप आएंगे तो प्रोजेक्टर कहाँ पे जाएगा. आपके आधे घंटे को गैप था केवल टीम में.
सर यहाँ पे काफी दिक्कत हो रहा है सेशन बी में.
So this shows the different kind of enzymes produced by genetically engineered material used for the treatment of cadmium, toluene, mercury, heavy and other materials in real environmental samples.
अमन और ये वाले में ये लोग क्या करेंगे?
So finally,
अभी तो गए थे वीडियो रन करने के बाद.
So
वीडियो रन कर देंगे हम. क्वेश्चन आंसर
microorganisms is much successful compared to the conventional method. However, for real applications, it is required further research on the complex environment and real environment.
ये 4 वीक
real environment. So I'm suggesting that still more research is needed.
यहाँ पे इसको क्या हो रहा है ना,
to field apply such kind of genetically modified organisms. Okay,
thank you, Satish. Thank you, Satish. You hold on. Yeah, you hold on. We'll have another presentation and we'll have a
हमारे पास
panel
प्रोजेक्टर
discussion.
नहीं है, होटल वालों ने ये बोला है कि हमारे पास तो प्रोजेक्टर नहीं है। अगर आप हाँ बोलोगे तो आधा घंटा प्रोजेक्टर अवेलेबल हो जाएगा ऑन रेंट। तो ये डिसीजन लेना है।
Professor Pramjit, there is a lot of noise in the background. Someone is talking. Can you just tell him not to talk?
मतलब
We can't hear anything.
मैं ये तो कर रहा हूँ ना, पहले तो हम ऊपर से जीत भैया कहना है कि एक बार प्रोजेक्टर बंद होता है 2-3 मिनट छोड़ दो उसको, 2 मिनट 3 मिनट टॉकिंग बंद हो जाएगा उनका।
छोड़ दो, मतलब 2-3 मिनट
छोड़ दो, क्योंकि उसका लेंस होता है जो बार बार रहता है, ऐसे कुछ बता रहे हैं हम, क्योंकि अभी चलते हुए तो 2-3 मिनट बीच में बंद करूँ तो प्रेजेंटेशन बंद है।
स्पीकर जो है स्पीकर ने रजिस्टर किया था, अगर वो उनको देना।
बाकी ये लिस्ट ऑफ 35 पार्टिसिपेंट है, उनके बदल दिया और किसी को
एक है केबी भूषण मिश्रा का एक स्टूडेंट है, उनके नाम पे एक सर्टिफिकेट जाएगा।
केबी मिश्रा कहाँ गया है? नहीं,
पता नहीं, वो बोल रहे थे।
नाम तो नाम दिया है, केबी मिश्रा ने नाम दिया।
हाँ, उन्होंने नाम दिया है।
कॉमा लगेगा क्या? कॉमा लगेगा ना डिपार्टमेंट का एक्टिविटी के लिए। देयर शुड बी कॉमा डिफरेंस। ब्लैंक सर्टिफिकेट साइन करवा के रखो ना, सब कुछ। हाँ, तो ब्लैंक रखे रहना, क्योंकि लास्ट मिनट पे क्या होता है ना, कुछ सर्टिफिकेट आपको तुरंत बना के देने होंगे, क्योंकि स्पीकर भी मांगते हैं सर्टिफिकेट। हाँ, उनका तो अभी बना लिया है। नहीं, सबको बना नहीं, लेकिन अगर जरूरत पड़ेगी तो नहीं पर ऐसे
सुजीत भैया को बुलाना ऊपर से जरा।
बॉडी है कृति टंडन आई है,
नहीं आई शायद, वो अपने नहीं, वो एक दिन आने के लिए बोले थे।
पार्टिसिपेंट का बना दो, ये वॉलंटियर को वैसे भी जाने वाला है।
अच्छा, नहीं एक्चुअली इसका अवार्ड है ना, इसलिए पूछ रहा हूँ मैं।
वो शायद आने वाली थी अपनी हस्बैंड के साथ एक दिन।
लेकिन ये बुलाया गया है कि ये
इसका क्यों बनाया हो? ये तो मुझे पार्टी नहीं, रीजन दीजिए, इसे बोलो ना, ये वो वाला है ना। देखो
देखो सर, मुझे ये लिस्ट दी गई है, इसका बना दो। कुछ लोग हैं जो यहाँ प्रेजेंट है, कुछ लोग नहीं है, उनको
रिगार्डिंग द वीडियो हियर। ओके, तो देखिए आपको कंसीडर किया सर, मैंने आपको नाम पठवा दिया। एक्चुअली मुझे जो आपने बताया ना कि
बताइए, तो ये क्या बोलते हैं, हमारे पास और है क्या प्रोजेक्टर?
नहीं,
लैब में
तो मैंने फोटो वाला नहीं भेजा।
मतलब ब्लैक वाली क्या? ब्लैक वाला तो नहीं चलता, ब्लैक वाला चलता है। क्या Okay.
Another one is a dye, like rainwater, coming from various industries, like not only textile, plastic, food, and so many industries are dumping various chemicals into the environment. That dye also impacts the ecosystem
ये
as
लोग
well
को
as
10 बार
fighting
बोला जाता है
pollution
सब।
and impacting our climate. So we need a method, like people are currently what we are doing integrated methods, which are chemical or engineering solutions for developing, like
बार बार आ रहा है।
स्पीकर को डिस्कनेक्ट कर रहा है, जब वहां से बात
everyone
करेगा
has
क्लोज
more
और तुझे
advantage to
सुनना
this
ही
approach.
नहीं
So
या म्यूट
to learn from the nature, it's already for
सबको बंद कर दो,
electric process,
डिस्टर्ब करेगा बाद में।
mechanism to tackle this challenge. So that's where we thought of combining with the chemical with this natural process, like we can get these microbes. And
Sir, you got the internet, sir? You got the internet?
I think
हो गया सर, आप काम कर रहे हैं? नहीं नहीं सर, आप नॉर्मल है। ओके।
क्या क्या हुआ सुजीत भैया, आप मुझे ये बताओ प्रॉब्लम इज विद द प्रोजेक्टर और क्या, लेकिन इनके पास प्रोजेक्टर नहीं है और बोल रहे हैं कि रेंट लेना पड़ेगा। अभी बताना पड़ेगा। ये प्रोजेक्टर फोकस नहीं करेगी, वायर का दिक्कत है, वायर ये पावर अप डाउन
कहाँ से पावर चाहिए आपको? बोल रहे हैं पावर का तो हर जगह होता है।
ऐसे भी फ्लक्चुएशन है इस कमरे में।
ये भी तो मैं कह रहा हूँ ना, पावर को वो बोल रहे हैं कि पावर को होता तो हर जगह होता है, यहाँ क्यों?
मेरा 99% प्रोजेक्टर में कोई दिक्कत नहीं है, मैं सच बता रहा हूँ, क्योंकि तुम्हारे में फ्लक्चुएशन नहीं है अभी, क्यों कल से
ये मैं कह रहा हूँ,
में भी हो रही है।
उन्होंने प्रोजेक्टर रिप्लेस किया है, नहीं किया ना, तो कैसे कर सकते हैं लोग? कैसे हो सकता प्रोजेक्टर
प्रोजेक्टर इशू होता तो इतना भी नहीं चलेगा। जो केबल का भी इशू, केबल अभी
हमारे पास बदलने के बाद वही इशू है, ऐसा नहीं है।
So what we started doing is like we are developing a high value-added polymer. With incorporating others like
यूज कर दो, आवाज आ रहा होगा।
We are making composition. I can't tell you like we can also develop that composition using artificial intelligence methods or computational solutions. So we are taking this lignin base from the industry, and the lignin needs to be modified. Like you can put any. Toxic compared to the native product. So still further, we are doing much research on this topic. So the overall, this is the solution we have developed. Like taking from pulp paper industry and convert that with some of the polymers to make it like kind of hydrogel or like high density polymers that can absorb this dyes and water process. Once it's absorbed, this microbes acts on cleaning them into lower molecular weight products that's much more less toxic compared to this native products. So here, what I was telling, like we are doing a like variation, like we can tailor-made generation of this material. So depends on what kind of, like here methylene blue, we can do this polymer composition as well as enzymes, whichever you want. You can make the tailor-made to treat, and also you can scale it up how much you really want. You can do this mathematical modeling and gain this
knowledge based on that, you can optimize the system. So overall, is this process of this hybrid solution provide a value addition to the industry like pulp paper industry and utilize the agro waste as well as this bioabsorbent used for remedial application. We are developing the green processes as a microbial degradation process that used for various effluent treatment from the industries. And this also we are working towards more zero waste policies like zero waste bioaccumulation potential. This technology is having that kind of advantage over it. And also this eco-friendly and non-toxic chemicals are generated is really greater advantage, and we can discuss more in detail. And also finally, we didn't do much techno-chemical analysis, but still it's coming from a waste, and we are like conceptualizing as like it's going to have more economical solution to the industry to handle these issues. With that, I thank once again thank CSIR and our team, CSIR idea, and thank you so much, and thank the chair for this opportunity. Thank you.
So, hello everyone.
Hello, thank you, Dr. R. Parthasarthi. Can Dr. Steve, please?
Yes, our next speaker is Amy Tan from University of Hong Kong, and she'll be giving a talk on the role of sulfur-reducing bacteria on granular activated carbon surface in anaerobic fluidized bed membrane bioreactor. And I think, Amy, they will be sharing your screen, and here you go. Yeah.
Try to do that. Oh, I think the PowerPoint is already shared by the organizer, so I think I'll use that. First of all, hi everyone, and thank you to Professor Samir and Professor Pranasar for your kind invitation to this conference. My name is Dr. Amy Tan, and I'm a research assistant professor from the University of Hong Kong. Today, I will be sharing with you on our findings on the role of sulfur-reducing bacteria on granular activated carbon surface in a new type of technology called the anaerobic fluidized bed membrane bioreactor, or AFMBR in short. Next slide, please. So as we may all be aware, the conventional wastewater treatment actually uses aerobic processes, which is very energy intensive, and in fact it accounts for about 40 to 60% of the total energy consumption in a wastewater treatment plant. And alongside with that, due to the fact that it's aerobic, it also degrades all the carbon compounds into CO2, so this leads to very high CO2 emission as well. So we've been looking at ways how to make this wastewater treatment process more energy effective, as well as reduce CO2 emission. Next slide, please. Therefore, we have been working with our partners in Inha University in Korea, Stanford University in US, as well as Imperial College UK. Specifically, we are looking at this bioreactor called AFMBR, and in the picture you can see a pilot plant trial of this reactor at Bukcheong wastewater treatment plant in South Korea. And this reactor is actually a membrane bioreactor, and it's also operated under anaerobic condition. And because of this setup, it's able to directly treat wastewater sewage directly, and also, we have added in electrically conductive granular activated carbon for membrane scouring, and it also acts as a biocarrier for the formation of biofilm. So this actually allows us to achieve very high solid retention rate, which allows us to apply this technology directly for sewage treatment. And based on the results of this pilot plant test, you can see that the COD removal is very good, 80% COD removal within a very short period of 8-hour HRT. On top of that, the methane yield represents a 50% enhancement when it's compared to the conventional sewage treatment processes. And overall, because of the methane production, it actually allows this treatment process to be energy positive. So what is interesting here is that this anaerobic treatment process is actually showing even better results compared to the conventional process and is able to overcome classical reaction kinetics in terms of pollutant removal as well as methane production. Next slide, please. So this actually is a very intriguing phenomenon to us, and we started to look at the microorganisms dynamics that's in this process to try to understand what is responsible for the enhancement. So based on various studies of various of this type of bioreactors, we noticed that it is very common for several types of microorganisms to form a biofilm on the GAC. Specifically, what we typically find is syntrops at about 5 to 10%, and we also find a special type of microorganism called exoelectrogen, such as Geobacter, that can be present at up to 5%, and also electrotrophic methanogen, such as methonotrix, that can be anywhere between 5 to 40% of the archaea population. And together, they actually form a very symbiotic community to enable the degradations of sewage organics, such as propionate, into acetate. And typically, in a anaerobic process, acetate would be taken up by methonotrix for methane production. But because of the presence of GAC, acetate can actually be taken up by Geobacter, which is the microorganism in green, and Geobacter actually oxidizes acetate and produces electrons directly, and these electrons can be conducted through the GAC material and be taken up by methonotrix for methane production. So under this kind of electron-induced process, the pathways in blue as well as in black are activated for the direct reduction of CO2 into methane. And this actually represents a kinetics that is 10 times faster than the acetate reduction to methane. So this actually explains the phenomena explains the good results that we saw with the reactor performance. Next slide, please. So we also started to look at another similar pilot plant setup at Stanford University at the Cortical Resource Recovery Center. So this setup actually treats municipal wastewater from Stanford campus directly. And because of that, the concentration of the influent COD is a little bit higher compared to the sewage. However, in terms of the COD removal and methane production, it is still performing at the very high levels that we saw earlier. And also, the influent sulfate and effluent sulfate removal is minimal, and the conversion to effluent sulfide is minimal as well. So we wanted to look at how the high COD concentration is influencing the microbial dynamics. Therefore, we sampled the biomass at 390 days, and we perform a genomic sequencing. Next slide, please. So basically, this is an overview of the methodology that we employed. We took the biofilm samples from the GAC, performed DNA extraction, followed by metagenomics and metatranscriptomics sequencing, and bioinformatics analysis. Next slide, please. So this is the results of the metagenomics and metatranscriptomics sequencing, just purely the abundance of the microbes as well as the microbial composition. And straight away, something very interesting popped up to us that was unexpected from our previous observation. For example, methonotrix is very high in abundance. However, its activity, which is indicated by the bar in orange, is very, very high as well. So this indicates that this electrotrophic methonogen is dominant and very active. Geobacter, which is an exoelectrogen, is also dominant and active as well. However, unlike our previous observation where synthropes are in large population, in this particular sample, we saw that the synthrope population, overall population, is very low at about 2%, and the activity is also very low as well, below 0.2%. And the other interesting phenomena we observe is these sulfur-reducing bacteria. They have a combined activity of 1.5%. So this is something that's very unique to us that we have never seen before in our previous sample. Next slide, please. So therefore, one of our first research question is, what is the role of SRB on GAC? Next slide, please. So from the metagenomics results, we recovered the genome bean and started to look at the pathways of one of the dominant SRB that we found in our sample, which is the sulfuro-vibro. And what we found is that this SRB has all the sulfate reduction process for assimilatory sulfate reduction, dissimilatory sulfate reduction and oxidation, as well as the SOX system. And we also observed that this particular SRB is able to oxidize propionate into acetate. And it also contains an enzyme called acetyl-CoA synthase that can further oxidize acetate into acetyl-CoA. And this is usually coupled with sulfate reduction. Next slide, please. So what this tells us is that in this reactor, even though the synthrope population is now decreased, but SRB is now replacing the synthropes, especially the propionate degrading synthropes, as the main propionate degrader. Because of that, it actually changes the dynamics of the microbial community. So now SRB is supplying the acetate to Geobacter, which can then oxidize the acetate for electrons production, and then this is taken up by methanogen for methane production. However, one of the interesting questions that we are currently investigating is the propionate oxidation to acetate is typically coupled with sulfate reduction so that the electrons that are produced can be taken up with sulfate as an electron acceptor. However, in this case, in the earlier data that I showed you, we do not see a lot of sulfate reduction happening. So therefore, one of the questions is, could SRB also then export all these electrons, and these electrons are then conducted by the GAC surface for methane production? So this is one of the questions that we are looking at right now. So in our future work, we will be investigating whether there are any extracellular electron transport proteins in SRB. And with that, how does this change the interactions between SRB and its other microbial partners on the GAC? So what is the interaction between this new SRB EET process and carbon as well as methanogenesis metabolism? And with that, I would like to also thank my collaborators from Korea, UK, and US. Thank you.
We'll come back on the panel discussion later. So our now next speaker or last speaker of this panel is Dr. Poheng Lee Henry, who is a senior lecturer at Imperial College London, and he will be giving a talk on non-aerobic respiration coupled with methanogenesis in efficient microaeration anaerobic digestion. And you will probably coming to share your screen, maybe.
Yep. Hi. Okay. Thank you for the panel's invitations. I would like to talk about, there's one new respiration which is observed in anaerobic digestion. So the topic is called anaerobic respirations with methanogens in microaeration digestion systems. So this work was also worked with Professor Cornell, one of our panel members. They're here at Hawaii. So next slide, please. So this outline, we're going to talk about what is anaerobic respirations and talk about reactor operations, and what is metabolism analysis in that. Okay, next please. Okay, so in UK we are looking at the net zero greenhouse gas emission by 2050. So a lot of energy efficient conversion will be increased. That's our target. Next, please. So anaerobic digestion is one of well-developed systems in industries, in academia, and we would like to seek that the currently only 0.6% of energy outputs from the anaerobic digestion. So we would like to think if we can enhance this efficiency of producing methanes. Next, please. So the limitation is that the current anaerobic digestion is still working on classical limitation. Can we surpass the classical respirations in micros? Next, please. So this is a general aerobic and anaerobic respirations. You can look at the oxygen levels. So one is above 1% that we call aerobic respiration, but less than 1% we'll call anaerobic respirations. Next, please, or fermentation. So we found that there's one metabolism is really ancient and not very common to see in our universe or in our Earth. It's we so-called anaerobic respiration. It's between anaerobic fermentation and aerobic. So there's a level between 1% to 0.001% of airs. Next, please. And this air actions level happen to be associated with methanogens. So we are thinking about if we can have lived actions and we can have anaerobic respiration to enhance efficiency of hydrolysis and couple with the methanogens, would that be a good combination overcome the classical anaerobic digestions? Next, please. In that case, we found that in aerobic respiration in electron transfer chains, the only complex 3 that acts the action into water is cyclochrome C. Next, please. And in ancient alternative oxidations, complex 3, next, please, is we call cytochrome BD, this enzyme. Next, please. And this has a highly action affinity. So we look at a few already sequenced AD systems. We look at the whole genomes, and we look at cyclone BD oxidates. It did show that there is a cyclone BD oxidates in all the samples we found, but it's a little bit less than high. And we also did a little bit resequence other lab scale of microaeration of their sequence, and we found that with a little bit more actions than cyclone BD oxidates, the genes is highly expressed. Next, please. So we got this kind of hypothesis. If we can have one special microbes that they can degrade cellulose biomass with microaeration in terms of anaerobic and anaerobic respiration switch on and off at the same time, that we can have a better removal of cellulose biomass, lignocellulose cells biomass, and produce more of VFA. But at the same time, the VFA can pass to methanogens, produce more methane. So they kind of break down this classical limitation on hydrolysis and methanogenesis. Next, please. Next, please. So we want to produce this boundary by putting a little bit action in the system. Next, please. So this was done by Professor Connell's group, that they did microaerations. The intermediate microaeration operations strategy is 25 millivolt of the ORP under above anaerobic baseline. So they find out this operation did a pretty good methane yield. Next, please. So in that case, at 5 grams VS per liter per day, the methane yield is 3 times with microaerations. And we could also see that on the carbon balance, we have a pretty good carbon recovery into methane. Next, please. So this figure is a little busy, but I just want to highlight that with this microaeration at high organic loading rate at 5 gram vs per liter per day, the pH can be maintained in really good around 7, but without microaeration, the pH is a little bit lower, around 4. And the VFA is maintained in a good range for the system. It means that this microaeration did a pretty good job in balance pH and VFA. So we can see that in this figure we did 6-ness RNA. The most abundance on the system is protein filings. So this looks like the first one. It looks like this one has some anaerobic respiration functions. Next, please. And also we found that methanol are dominant microbes that can use hydrogen acetate. Next, please. So this figure is a little busy. We did find out 2 major microbes, genome being so-called protein filin species and methanogens. And I want to highlight the more key finding is that, next please. We define anaerobic respiration, that is red colors, that they use cyclone BD in a system. And also they produce more hydrolysis enzyme, special cellulose biomass enzyme. Okay, next please. And at the same time, they also encode with the enzyme. The green color is reactive oxygen species detoxification metabolisms, which means that they can kind of defense the detoxification of oxygens. And then this protein filins in the blue columns is they can do anaerobic respirations or anaerobic fermentation that produce acetate. So the acetate will give into methanogens. Next, please. So that they can do produce methanogens. Next, please. So now in our current ideas that we are actually investigating this cyclone BD oxidase with Professor Amy Tans at Hong Kong U and Professor Ilya at in Germany. So we look at this enzyme really unique, and we compare with Geobacillus, this thermal denitrifying that microbes. Next, please. And we find out this microbe has really high affinity to oxygens, and we hypothesize that this oxygen transfer rates or electron transfer rate should be really high. And we found out in cyclone C, it has a quantum tunneling effect in this aerobic respiration. So we hypothesize that this cyclone BD oxidase also have this quantum tunnelings, and we are usually use the microtheory to calculate is this quantum tunneling is also in this proteins. Next, please. So the closing remark is that this overlooked anaerobic respirations coupled with the methanogen is actually well abundance in our environment, but it's been overlooked. And if we use high rate of microaeration, we can enhance this function and produce more methanes, and hopefully we can reduce more of CO2 reductions, but turn into methane. Okay, so those are our collaborators in this study from Imperial, Hawaii, Hong Kong, Jacobs, and Michigan and Harper. But also we have one collaborator from Germany. Thank you so much. That's my talk for today. Thanks.
Dr. Lee, Puheng Lee. So we will now close this session of the panel. So we're going to go to a panel discussion. Joining me is Professor Paramjit Panesar. Together we will be doing a panel discussion. So I would like to request all the panel members, please be ready, and we'll start the panel discussion. It will last maybe about 20-25 minutes so on. So we'll probably wait for there are any questions from the audience or any other questions from the chat. We'll wait. Professor Panesar, are you ready there?
Yes, hello. Hello, am I audible?
Yes, so shall we start the panel discussion now?
Yes, we are ready with that.
So there's any...
Yeah, Professor Smith, you can start and we will continue with that. Okay, just one query from the Professor Hector. That is, what are the upscale challenges of hydrothermal liquefaction? Can you elaborate on that? Upscale challenges.
This is
Okay.
for whom? Okay, for maybe better to.
This is for
Okay.
Professor Hector.
Okay. Well, in terms of the hydrothermal processing, is the question?
Scale up, yeah. Question about scale up
Okay.
of the hydrothermal, yeah.
Okay. Well, thank you. Well, it's true. At this moment, the most important challenges for the hydrothermal pretreatment is try to pass for the batch mode operation to continuous operation for the pilot scale. So it's very important to try developing these strategies for using, for example, the severity parameter is an mathematical modeling in terms of using the operational conditions in terms of the temperatures and the pressure and time. It's important to considering the particle signs during the processing. But the most important at this moment is try to pass for the batch and the continuous using this kind of parameters as a strategy, the severity parameter.
Professor Hector. Another query is for Dr. S. Mohan. That is how zero waste is possible when we are talking of biorefinery. And we also know at some stage or other, some of the waste is always generated. And we always focus on the zero waste.
Thank you, Professor Pranad. You are absolutely correct that zero waste is at present not possible, but adapting biorefinery strategies try to integrate process in a systematic and an integrated way. We can recover the resources. That's what we are looking into. By taking biological waste, by integrating with two or three biological process, or two processes of biological nature and thermochemical process, we will be able to recover 90%. Apart from CO2 which is being generated from process, we can also recycle in the closed loop operation so that we can achieve near zero emissions from the process. So, however, this required, there are a lot of challenges to upscale this one because each biological process requires certain amount of retention times and conditions. So all integrations at a scale that is viable to look into technological feasibility as well as life cycle assessment, then we'll be able to tell the sustainability as well as its practical implementation. Thank you.
Okay, thank you, Dr. Venkatramohan. So the next query is for Dr. Satish Kumar. That is related with because his lecture was mainly focused on the GEM, that is genetically modified organisms. And can you elaborate on the policies of the GEM, particularly in China, and how there can be related to the agriculture applications and the real system that we're talking about? Because environment is also one of the topics that was elaborated by this Satish Kumar. But agriculture and environment, they are both they're very, very important. But if there are risks are there, then how we can control it? Because this is a burning issue in different countries. Europe has another policies, India has another policies. So you have been to India as well because you belong to India, and then you are also working in China. So can you just speak on that, on the policies of related to German real systems we have discussed? Professor Satish Kumar. Kindly unmute yourself.
Prof, one second, please. Hello? Hello?
Dr. Satish, just my question was Can you elaborate on the policies related to genetically engineered modified organisms with respect to agriculture and environment?
Yes, sir. Actually, we need there is no strict regulations because all are under research progress. So we need more regulations for real application proof, because in environment it pollutant contaminated in complex nature. So we need more regulations for this one.
Professor Satish Kumar, I have one question regarding when we apply the genetically engineered microorganisms in real field conditions, how it will influence by native species? Is there any kind of innovation, and how adaptability will be there? Hello, Dr. Satish Kumar.
Madam, please. Yes,
are you able to hear? So Dr. Satish Kumar, I have one question. That's regarding are you able to hear?
Yes, I hear.
Yes. Using modified organism germs in real field condition, how effective they can survive in terms of is there any kind of innovation?
Little background noise. I couldn't hear you.
So what are the challenges when we are using genetically engineered organism in real field condition? Can you throw light on that?
Yes, sir. Yes, Prof. Actually, if you're using the real field, so the mutant will be regularly continued. So it has some problem in the real application because of the mutant will not regularly continued. So the mutant deletion has been occurred for the real applications. So some we need to optimize the real environment condition. Hello. Also.
Yeah, I think some network issues there. Satish.
It's quite difficult.
Hello. Okay, no issue, we can move further. This question is for Dr. Amitan. That is, you have just discussed for the different bioreactor designing for the anaerobic microorganisms, basically, and self-producing bacteria. There are USB reactors as well, that is upflow anaerobic vapor reactors, then anaerobic vapor reactors. So, can you just compare, that is, which one can give better results, or depending on your more based on your studies? Because you had basically focused on the anaerobic fluidized bed reactor. So, are there any other reactors that can be used for this purpose, or this is the only you had screened based on your studies?
Hi, sorry, I didn't quite get the last part of the question. Could you repeat?
You had discussed about the anaerobic fluidized wet membrane bioreactor. Okay. So there are other reactors as well, that is AVR reactors we call them, anaerobic baffled reactors. Then there are UASB reactors as well. So have you worked on those reactors as well, or you have worked on AFMBR only?
So personally, for myself, I've only worked on AFMBR reactors for direct sewage treatment, and I've also worked on anaerobic digestion reactors for sludge treatment.
Okay, Dr. Ami, I have one question, actually, clarification. Basically, you are working on a direct electron transfer by using conductive materials. Can you throw light on how it will be affecting syntropy with the direct electron transfer? Because you have done excellent work on that. Just to get an idea how we can use the T-selectors.
Right. So from our observation of this conductive material, the microbial community on this conductive material GAC, it is very often that we see that it actually enhances the growth and dominance of syntrope population. In particular, we see propionate degrading syntropes that are enhanced on this GAC. So as we know, actually, the syntropes, they tend to be in the lower abundance in regular anaerobic digesters. So definitely the addition of this kind of conductive material is actually beneficial in terms of enriching for this kind of syntropes and also facilitating the degradation of propionate to acetate and then acetate to electrons for methane production. However, in also the recent study that we looked at, the reactor in Stanford, we also see that if the propionate degrading syntropes are not in high abundance, then it looks like another group of microorganisms, the sulfur-reducing bacteria, can be enriched to take over this function. So it looks like with the GAC, the presence of GAC, it can help in the overall enhancement of pollutant removal as well as methane production and as well as increasing the yield of methane, either through enhancing syntrope population or other forms of propionate degrading microorganisms. So this is what we have observed so far, but of course there's still much to be learned from this kind of bioreactors. So we are still trying to understand more, as well as also the roles of other types of microorganisms like the fermenters, the hydrolysis. Thank you.
So another query is for Parthasarathy. That is related to the computation methods. That is, what software can be used and in designing the hybrid matrices particularly. What is their cost involved, and whether there are some copyright issues are there. So if we can elaborate on that.
Thank you, sir. And actually, this is a very pertaining question. So here I'll answer it in two segments. First, I'd like to mention how this computational approach is going to help in designing or particularly first understanding how it's worked, understanding the mechanism, and how it's going to be develop something new, which is to overcome some efficiency or robust methods. Like if you want to, probably when we are using it, like we are using some fast solvents like acids or base, how we can incorporate the biocompacted materials like cellulose, collagen, chitosan, or mineral derivatives or colloids. So that we can take them and how we can modify or tailor them made to handle or how to degrade this pH or several other contaminants and chemicals. So we are using several computational approaches on this, like quantum mechanics methods to understand the mechanism. And if you want to develop how this system works, so we use like multiple resolution methods like molecular dynamics simulation or system biology-based approaches. And later point, sometimes like we are also using CFD, like fluid dynamics models to understand the kinetics and mechanisms. So there are several methods are involved. The computational approaches are like going to be advantage into collect information. Like Morning also said, the data science methods, like we are collecting the methods depends on what target, what product, or like what process you want to make and your efficiency. And based on that, you can develop a database that can guide you in terms of how to make an efficient process. So that way, it's going to reduce, like your all the preliminary work has been done. You need to do the optimization methods. That's what preferably we want to enable as a system, as a predictive method, so you can go for your real testing at your lab level or field level. So that way, it's going to impact more on your reduce, reuse, and recovery. So all those things will be much more like approaches. That's what we can refine them. So this process will enable to reduce the intensive resources for developing your technological solution. in the sense. Like for a microbial, we have a PAH that several microbes can degrade. You can sequence them, and the sequence, I think Dr. Palnivel also mentioned about it, like how genetically modified organisms are developed. Similar to that, you can make some kind of sequences that can efficiently degrade PAH. Because she mentioned about electron transfer mechanism, like some of the single electron transfer mechanism and so on. Several mechanisms are also you can incorporate because how it's going to be involved and degrade the particular contaminants or the pollutants. So that way, computational approach is having a lot of advantage to understand the mechanism as well as upgrade this process or fine tune the process or like scaling up the desired process for your applications. Yeah, thank you. So there are several softwares that are open sources or like commercial sources. I don't want to go into more detail on it. Like depends on your like level of problem, you can use these softwares efficiently. Yeah. Thank you.
Thank you, Dr. Pratap. So I have, Dr. Lee, I have one question. So you have talked about microaeration, which along with Professor Samir you have done. It's a very interesting work, and you got very good results with different metabolic approach, looking into EFA without EFA production. But I have one query. When you take micro aeration, what happens to strict methogens, which are also very much relevant for methane production?
Oh, so if I recall your question correctly, you mentioned about methanogens, how the methanogen will resist the actions. Am I correct?
Microaerobic aeration, how methanogenic survival, strict methanogenesis survival, methanogenic bacteria.
Yeah, from the culture we saw in the microbes, especially in archaea methanogens, the only dominant methanogens that they are encoded with detoxification of actions species. If those are strictly anaerobic, maybe it's not shown in this kind of community. But this ROS detoxification methanogens, actually they are actually doing pretty well producing methanes. Doesn't has big difference between those fact extremely anaerobic condition methanogens. That's what we observed. Okay, thank you.
There are some queries from the audience as well, Professor Moholkar.
In many microbial processes for wastewater treatment, it has often been seen that some of the intermediate metabolites are more toxic than the original pollutants. So in that case, like, what is the best strategy to adopt, like to avoid that kind of situation, or is there any new metabolic engineering being developed to avoid that particular pathway that metabolites are more toxic than original pollutant?
It's an interesting question, Professor. I will like to say one example when we are dealing with textile dyes, particularly azo dyes. So we require microenvironment to break down the molecule. So the best example here is first we'll give the aerobic conditions where the azo dye molecules will form into amines where anaerobic conditions cannot take amines. But in the when we send it to aerobic process, then amines get reduced. So by integrating multiple metabolic approach like aerobic, anaerobic, anoxic, aerobic, basically by using sequential batch reactor, to certain extents we can remove the pollutants. This is the example where I have studied. So similar kind of things for toxic molecules, there are approaches I have seen.
Because even like our environmental constraints, like the norms that are given are on the basis of COD, POD values, but they don't go for individual metabolites or toxicity. So what I wanted to point is that is there any necessity for prescribing a toxicity limit as well, other than POD, COD values?
Yeah, that is actually that is going on. But Doctor, can you
biggest fellow from actually it's very very, very valid concern. Actually, that is going to also kill these microorganisms, like whatever the product is going to generate. So, Lee, I think you have Dr. Lee, he also mentioned pretty much similar approaches, non-aerobic process followed by several methods to avoid this intermediate species. But if you, like yesterday, if you remember, there are also, they are developing biosensor. If we, anything can be, this is what, but overall, we need to understand different process conditions. It's going to work or not. And that limit should be like, depends on how much feed, like depends on how much going to get in and getting going out. If you control that, then probably you can reduce the formation. That's the fundamentals.
I think there is another question from the audience.
You can set the limit.
Mixed cultures might be like mixed cultures rather than isolated cultures might be.
Sir, I'm Dr. Bossa from ICL InFed. My question is to Dr. Parthasarathy, sir. So my query is, sir, is it possible to functionalize lignin-like hydrophilic branch-chain polymer with some polar groups containing both positively charged, like groups, and some negatively charged simultaneously, so that it can absorb both anionic and cationic dyes from the effluent water? And another one, is it possible to functionalize it with some non-ionic hydrophobic so that it can attract dispersed dye-like molecules? A lot of textile industries are using dispersed dye for dyeing the polyester kind of textile garments actually. This is the one major problem of the effluent also. But we need some hydrophobic kind of groups for catching this kind of the dyes from the effluent. So is it possible to functionalize lignin like hydrophilic branch chain with some hydrophobic functional groups so that it can attract this kind of the molecule from the wastewater?
Thank you for this excellent question. So, for first one, like there is a saying, like you can get anything out of lignin except money. So you can tune the lignin the way we want. So we have also made some kind of example of like one of our PNAS work. We take the lignin, convert it into this amination process into ionic liquids. If I make ionic liquids like cations, and also we also form the anions also, like various combinations we can make. From it has a SGS unit, right, like hydrophilic, but that's a hydrophobicity is very important for the absorption. So definitely we can fine tune them, but that process also requires a lot of economically viable process we need to develop. That's the catalytically people are started working on like MOF, geolites and attaching with lignin and how it's going to cleave these dyes and all, like there are several efforts going on in the literature. But how it's going to be practically incorruptible, but they have demonstrated it. The efficiency also high, like more sometimes 80 or 70 to 90%. Some of them they have shown the efficiency to break this degraded product, like from dyes, your industrial dyes. But it's very challenging, and I think this is what you can definitely make cation and anion and simultaneously. I've seen a couple of papers, maybe I can share it with you. But other than that, what is your another question?
Hydrophobic function.
Yes, it's there, already there. That's why we started using this. Yes. Thank
you. Thank you, Dr. Park. Any queries from Professor Smith, please, if you can, if you have any questions from the panelists. Professor Smith. Professor Smith, yes, sir.
No, I don't have much question because I want to ask some question, but then has already been asked. But Hector, I have a quick question for you. In the high solid, did you look at the mass transfer limitation? Because if you look at higher concentration of solid, mass transfer may become an issue. Did you have any comment on that side?
Yeah, Professor Samir, it's true. We have many problems in terms of the mass transfers. In terms of, for example, the viscosity during the processing in any stage. For the hydrothermal processing, in this case of biomass fractionation or the enzymatic hydrolysis or the fermentation, for example. We need to try to develop very well the designs of the bioreactors for try to improve this phenomenal situation in specific for the viscosity. It's true that we have a delimitation in this part. We have inhibition with different issues. For example, in the enzymatic hydrolysis, we have the absorption of the enzymes onto the lignin, for example, when we have a high solid loading. So depending, but it's very, very important to try to study very well the situation of the mass transfer. In this case, the viscosity. Yeah, this is true.
For the Satish Kumar, I have a question. I think also same question was raised by Mohan also about the genetically modified organisms in the environment. That could be a big challenge in terms of policy, in terms of environmental issue, because GMO is a big issue. So there could be a debate on putting genetically engineered microorganism in the environment. So engineer bioreactor may be okay, you can probably control it nicely before you discharge it, but in the field application, maybe there could be some policy issue or some other issue. Did you have any comment on that side? You're muted. I can't hear you. I can't hear you.
Hello. Kindly unmute Dr. Satish Kumar. Kindly unmute yourself. Hello. Dr. Satish, you are mute. Yes.
So I have one question for Dr. Tan. Quick question for you. You mentioned about using SRB. So when you use SRB, you may read.
Hello.
Sorry, I lost. I got frozen. So did you get my question, by the way?
Sorry, Professor Samir, I didn't get it. Could you repeat it
My
again?
question was, because when you have sulfate, I know it's sulfate, mostly sulfate form, right? So when you have sulfate in the system, in the sulfate reduction, it will consume electron donor. So it could be hydrogen, it could be acetate. So when it consumes electron donor from substrate, so you will reduce the amount of electron donor available for methane production, acetate and hydrogen. So you may probably reduce methane production overall. But you mentioned that you enhance methane production. So I was a little bit contradictory points. I was just trying to clarify that part from your side.
Right, yeah. So that is something that's very interesting and unexpected to us, because in terms of the methane production performance, it was actually maintained. So we started to wonder then what is the SRB doing. So at first we thought that SRB was reducing the sulfate, using that as an electron acceptor. But if you may recall from the sulfate data, there was not a lot of sulfate reduction. Very little hydrogen sulfide were actually formed. So this actually indicates to us that the electrons are probably going to somewhere else. And one of our hypotheses is that the electrons could be exported by the SRB and get conducted through the GAC to methanogens for methane production in order to continue help enhancing that, so that can explain why we are not seeing a reduction in sulfate amount or production of sulfite, but we are still seeing the very good methane production results. So that's why one of our next step is actually to check if this hypothesis is true by looking into the genome of this SRB to see if we can find any extracellular electron transport genes that could help explain this, and I'll later on confirm that through in gene expression results.
Point, because also Professor Professor Pram also asked about different reactor system, but I know your reactor has a special function because you are using GSC to reduce the Pauling propensity. So that's why you use GSC and membrane bioreactor. So that's the function of the GSC, in my opinion, right?
Yes, so both for membrane scouring and also as a biocarrier.
That's right. So that's a special function. So that's why it may be difficult to compare with USB or other type of reactor in that particular sense, but very interesting. Yes. So Henry, I don't have any question for you because we work together and then we have discussed a lot. So excellent presentation. Yeah.
Okay, thanks.
Yeah. So I think Professor Pram said I am done with my few queries. So I think, shall we close it this one and head to the next rapid fire presentation?
Okay, thank you, Professor Smith. Now we will be having the contributed papers. There are 7 contributed papers, what we call it rapid fire session. So,
I have a small comment regarding the point of mass transfer raised by Professor Samir Khanal. Now, in microbial systems, am I audible? Yes. Am I audible? Yeah. I have a small comment regarding the mass transfer issues raised by you. Now, in case of microbial systems, the nature of convection that we induced in the system is very important. There are 2 types of convection. One is macro convection, which is usually the stirring or turbulence or basically stirring we provide. But that kind of convection is not suitable for enhancing mass transfer in cells, microbial cells, because those are all micro levels. So what we need to introduce is convection on that level, micro scale, so as to enhance the mass transfer in that kind of system. So if you remember, like a few years ago, there was one of my students' thesis had come to it that we had used ultrasound for enhancing system is not important. At what time scale and what spatial scale we use, the energy is also important. And second comment I have regarding what we were discussing, like what is the role of that immobilized support for enhancing microbial processes. There are 2 roles. First is that you increase the cell density to the system by immobilizing system, and second is that you enhance the interaction between substrate and microbial cell. So if the adsorbent, if the immobilized support is also acting as an adsorbent for the dye, obviously it will give double enhancement because it will concentrate the pollutant in a relatively tight system, which will enhance its transport into the microbial cells. So these 2 factors, like adsorption and convection, are quite important in enhancing microbial processes.
Okay, thank you, Professor Mohan. So, actually, we are running short of time, so we're closing this discussion session. So, next, we have the contribute papers. 7 papers are there, and panelists will act as the judges for the evaluation of these papers. So, they must have received the evaluation sheets with them. So, first, this lecture, that is a paper ready by Colonel Kumar. He is present physically, so he will be giving this lecture in digital mode. So kindly stick to the time. So there is 3 minutes. The time is there. Okay, thank you.
Okay, greetings everyone. I am Karan from IIT Guwahati. I am here to present my work. I am here to present my work on inhibition, finding out the inhibition mechanism of the solventogenic enzyme present due to the inhibitors present in hydrolysis of. So already said by the expert lectures. So I want to highlight that the conversion of biomass to fuel requires the 3 steps, which is pre-treatment, detoxification, and fermentation. In pre-treatment step, many fermentative inhibitors are produced. So, and that has not been explored much in the scientific literature. So, I have aim to study this inhibition mechanism of the solventogenic enzyme, which is alcohol aldehyde dehydrogenase, caused by these compounds present in hydrolysis of cellular biomass. What I have done in this study, I have identified the key enzymes, key solventogenic enzymes, then modeled their structure using homology modeling tools, then energy minimization and other steps, then followed by the identification of inhibitors and the modeling their structure using DFT simulation methods, and then further the molecular dynamic simulations to understand their structure, structure analysis and interactions. These are the softwares and tools used in this study. Coming to the result section, these are the identified key solventogenic enzymes which have been reported, and this is the structure. This is the structures of obtained after the homology modeling, and here we can find we can see that these are the structures that were generated using the DFT methods. We can see from this table that representative substrates had the higher binding affinity towards the substrate, towards the enzymes, and the inhibitors had the lower binding energy. So, because this suggests that they were inhibiting the substrate, they were inhibiting the enzymes. And coming further, this was further analyzed using the molecular dynamic simulation, and we understood the structures and other information regarding the interaction. In conclusion, we can say that this is this can. Oh, sorry. In conclusion, we can say that this study will further fuel the development of engineered enzymes and also support the future study for developing more robust and sustainable routes. I thank Prime Minister Sessions Fellowship for funding my research and permission facility of IIT Guwahati. I acknowledge my supervisor and my interns. Thank you all.
Well carried out work, Karan Kumar. And quick question is like, have you ever checked any competitive binding together?
Yes, sir. The competitive binding I have checked, sir.
You have checked it?
Yes, sir. I have not reported because there was a slight limitation. That's why I could not show the all results. But I have checked the competitive binding means first bounded the substrate and then bound the inhibitor and studied that was the test. In control, the enzyme and inhibitors were bounded first, and enzyme and substrate were bounded first. That was compared for analyzing the different interaction. What are the different changes in that?
Okay. Excellent. So, do we think that this competitive binding is going to change? What kind of changes you are expecting?
Yes, sir. I was expecting the different quantitative inhibitions, such as if a substrate is bound on the particular binding site, and then inhibitor is again going to that binding site, it means the competitive inhibition. But if the substrate and inhibitor is not competing against same binding site, it means
That's what I got.
Yes,
it want to bind the same side or different side?
Yes, that was also model, and that was repeatability, but I could not proceed.
Thank you.
Okay, so thank you, Karan. So the next participant that is Srishti Gupta, paper 76.
Good morning, everyone. I am Shrishti Gupta from CSIR, Indian Institute.
You guys can hear something? I can't hear anything.
Same here.
Same here. I see. I was going on and off. What's going on? First one, okay, but second one, I could not hear anything. I don't think this is a good platform. Actually, it has a lot of issues, challenges. Actually, I am surprised. Zoom is actually pretty good.
Yeah.
It's very late for you, Hector, right? I guess it must be midnight, after midnight, something like
Yes, it's 1 a.m.
Yeah, 1 a.m. Uh,
Yes,
you're okay. I mean,
it's
I
okay, no
know.
problem.
Henry, you are sleepy. I guess you can go back and sleep after this.
Yeah, it's true. But yeah, no problem.
फिर यहाँ से माइक्रोफोन लेने लगेगा तो वो नहीं लेगा ठीक है you can start
Good afternoon, everyone. We are
start
Shivani from CSI and Institute of Toxicology Research. Here I am going to present my research work based on green synthetic approach for bioabsorbent development, where we synthesize lignin-based absorbent for mercury and removal from industrial effluent. From the absorbent context, we have estimated that absorbent technology will rise at a CAGR of 6.18 percentage by 2026. We have already assumed that absorbent have emerged in various industries, for example, pharmaceuticals, textiles, solvent recovery, etc. Here we approached lignin-based absorbent for industrial effluent treatment as a waste-to-wealth approach, where we combine lignin with polyvinyl alcohol for absorbent development. From the industrial effluent treatment, we have considered mercury ion removal, where mercury ion is discharged into natural water bodies via coal-fired power generation, municipal waste combustors, dental preparations, etc. There are already available technologies for heavy metal ion removal, but as we know, metal ions can be converted into various forms but cannot be degraded to non-toxic forms. So absorption process has been considered as the most feasible and efficient method for metal ion removal. From the synthetic process, soda lignin has been graft polymerized with polyvinyl alcohol that has shown high swelling properties. From the results, the FTR data of from the results, the FTR data revealed that the successful cross-linking between soda lignin and polyvinyl alcohol. The broad peaks have reduced that shows the complete cross-linking between lignin and polyvinyl alcohol. From the absorption efficiency, the absorbent have shown 200 grams per gram of hydrogen water swelling capacity. From the cell microstructures, as compared to the control gel, the lignin-based absorbent have shown various ranges of pore sizes ranging from 20 microns to 40 microns due to the amorphous nature of absorbents. In conclusion, our main strategy was to successfully synthesize lignin PVA gel via fissile and one-pot synthesis mechanism that will prove as a sustainable and circular economy approach to fluid treatment. About 50% lignin has been successfully utilized for gel preparation. The absorbent have shown 90% removal within 1.5 hours. The absorbents are stable and can be reused up to 5 cycles and has proved to be a low cost and environmental alternative as compared to the currently available absorbent, that is polyvinyl alcohol gel. Thank you.
So, Sita, after 5 cycles usage, can you regenerate the absorbent for further use?
Actually, sir, the absorbent mechanical strength will be reduced because after soaking the cross linking will be disrupted due to after soaking multiple times. After five cycles, the efficiency will be reduced up to 50%. It has been observed.
Thank you.
Thank you, sir.
Okay, our next participant, that is paper number 86, Akila. Peter will play for this lecture, and I think she is online as well, so we can ask one question.
Hello everyone, my name is Akhila Nair and today I'm going to disinfection performance of method of nanomaterials for virus inactivation under the supervision of Professor Swatendra Pratap. So as per the reports from WHO, 1 in 3 people lack access to safe drinking water. Waterborne pathogens are considered to be a major reason for disease outbreaks in the world. And among the waterborne pathogens, enteric viruses are the viruses that are transmitted through a fecal-oral route, and they possess certain health hazards such as meningitis, encephalitis, and pneumonia. Various conventional methods have been employed such as thermal, UV, ozonation, and chlorination in order to reduce the concentration of enteric viruses in wastewater. However, these methods face certain drawbacks such as formation of disinfectant byproducts and higher energy consumption. Thus, there is a need for alternative disinfection methods with lower cost and less impact on the environment. So the purpose of our study is going to utilize electrochemical disinfection methods which employs the passage of electric current to nanomaterial-based surfaces. The nanomaterial surfaces which we have used is laser-induced graphene, which have higher conductivity and higher surface area. We have also doped these surfaces with certain metallic dopants such as TiO2 and Ti4O7, and we have checked the efficiency of these surfaces for virus inactivation. Coming to the methodology, preliminary batch setup experiment was carried out with LIG and doped LIG electrodes. So as you can see, this is a figure of the schematic illustration of batch setup wherein the electrodes were immersed into the MS2 solution. This MS2 solution is basically the phage solution, and MS2 is a model phage for enteric viruses. These electrodes are basically connected to the DC power supply. Following the batch setup experiment, filtration experiment was also carried out with LIG and doped LIG filters. This is a schematic illustration of the filtration experiment wherein these filters were placed in the filtration unit which is connected to a DC power supply. Coming to the results, in case of batch setup experiment, we have done a comparative analysis of LIG and doped LIG. The dopants used were TiO2 and Ti4O7. This is a percent inhibition graph and this is a log removal graph. And as you can see in this graph that the inhibition trend were as follows, wherein TiO2 LIG showed the maximum inhibition followed by Ti4O7 LIG and then LIG. The maximum removal of 6 log was achieved within 45 minutes of operation in case of TiO2 LIG. Coming to the filtration setup experiment, in this case as well, TiO2 LIG showed the maximum inhibition of 6 log at 2.5 volt, whereas the other filter showed 6 log removal at 10 volt. So in conclusion, I would like to say that the unique properties of nanomaterial, in our case laser-induced graphene, make them an excellent candidate for virus inactivation. Further, doping of this laser-induced graphene with certain dopants has enhanced the properties of nanomaterials for virus inactivation. Thus, the mentioned strategies will provide a highly efficient approach for the disinfection of viruses from wastewater. These are my references.
Hello? Yeah. So, interesting work. Can you comment on your system, how effective it can disinfect bacteria? You have worked on virus, how effective it will be bacteria?
So the effect is going to be kind of similar when it comes to bacteria as well. So colleagues from our lab has done with bacteria as well, and we have found similar kind of result in case of bacteria as well, for both gram-positive and gram-negative bacteria.
Okay, thank you.
Thank you, sir.
Okay, thank you. Next paper, that is number 87, methyl choline.
Water and wastewater treatment sector basically due to its requirement of less area, production of a high quality water, and facile mechanism of operation. There are basically 4 types of membranes based on their pore size: microfiltration membranes for the removal of suspended particles, ultrafiltration membranes for the removal of viruses and bacteria, nanofiltration membranes for the removal of trivalent ionic impurities, and RO membranes for the removal of monovalent ions. In this study, we will be focusing majorly on ultrafiltration membranes. To these membranes, first one is the fouling, that is the accumulation of particles and molecules on the membrane forming a gelatinous layer over the membrane surface which hampers the permeability of the membrane. And second is the selectivity permeability trade-off, that is if the water permeability of the membrane increases, its selective nature or the rejection of the membrane decreases drastically. Hence, we require certain modifications of the membranes so and a highly selective membrane to produce a higher quality of. So there are basically the method for the fabrication of asymmetric ultrafiltration membrane is done by non-solvent induced phase separation method. That is, by preparing this polymer solution, we pour it on the glass plate on a membrane casting machine, and the glass plate is then kept in the water bath for coagulation, and then we get an asymmetric membrane. However, for producing a symmetric membrane, this glass plate of the casted membrane is first kept inside a humidity chamber and then kept in the water bath for coagulation, and we get a dense sponge-like structure for the asymmetric membrane. Three different membranes were tested for pure water flux and BSA rejections by exposing for 3 different times. First is the asymmetric membrane, that is, the membrane was not exposed to any humidity. The second membrane was exposed to 5 minutes of humidity, and the third membrane was exposed to 10 minutes of humidity. And as we can see from the graph, the permeability of the second membrane that is exposed to 5 minutes of humidity gave a very high flux, which because of its finger-like structure of the pores, and the membrane with 10 minutes of humidity exposure gave a very low flux but a very high rejection, that is up to 53.18% because of its dense nature. Hence, humidity affects the nature of the pores of the membrane, and optimization of time of exposure to the humidity is required so as to prepare a porous and dense symmetric membrane to obtain a better permeable and selective membrane. Thank you.
Are you able to hear?
Hello. Yes, sir.
So, can you comment on reject? How much effective your system is handling the reject with reference to quantity?
So I had tested this membrane on a dead-end filtration system. So these results are for the dead-end filtration. So I haven't checked it for the reject. So these results for the BSA, that is the removal of the BSA. It would be more appropriate.
Okay. Thank you. Thank you.
Yeah. Thank you, sir.
So the next lecture that is apprenticeship by Manupati. That is number is 138. Kindly.
Hello everyone, I am Tarak Atmakuri, research scholar under the supervision of Dr. S. Venkat Monsal. Today I am going to present the collaborative research work of CSIR, ICT and CCMB, long-term surveillance for SARS-CoV-19 wastewater-based epidemiological studies. Why wastewater-based epidemiology? WBA an approach to potential to minimize the outbreak of the pandemic and also serve as the alarming for the future outbreaks. Our study area is the Hyderabad, including the STPs and lateral and main drains of the Nacharam, and also some lakes surrounding the Hyderabad and Telangana. The kits we have used a poison kit and a quagin kit, and genes we have identified as engine, EGene, and ORF1AB. So our first study is the STP surveillance. We have collected the 30 samples from the 14 inlets and 14 outlets of the 7 STPs and tested for the surveillance of the SARS-CoV-19 genes. All inlets of the STPs have detected the EGene, engine, and ORF1GB. Interestingly, outlets are not detected. So that shows the effectiveness of the STPs for the treatment of the wastewater. Apart from the STPs, we have also done the methodological approach method conducting the WB study, and we have considered the one selected drain and collect the samples from the 5 a.m. to 4 a.m. In 24 hours. RNA copies in the early morning hours, that is saying to 9 a.m. where the domestic activity is more prone, has shown the higher RNA copies. And these methodological study provide the information for the designing the sample protocols to improve the sensitivity and adapting the safety protocols to interpret the SARS-CoV-19 data. In addition to the methodological approach, we have conducted the long-term and long-term surveillance. We have collected the samples over the 1 year from July 2020 to May 2021. We have calculated our study area into 3 different zones based on the severity of the infection dynamics, red zone, green zone, and orange zones. And along with the longitudinal surveillance and long-term surveillance, the lake monitoring. In the lake monitoring, we have selected the 5 different lakes: Nizam Talab Lake, Uttaraj Lake, Idlabad, and Hussain Sagar. the lakes were more prone to the domestic activity shown the presence of the SARS-CoV-19 genes, whereas the lakes far away from the Hyderabad and more far away from the domestic activity, where the water is generally used for the farming, show the less and no detection of the SARS-CoV-19. Along with Telangana study, we have collected the different we have collected the samples from the different STPs of the Andhra Pradesh also, including the Visakhapatnam, Vijayawada, Kannur, etc. And we have collected the infection dynamics and RNA copies. These infection dynamics and number of infected people's infection rates are correlated with the number of positive cases of the Andhra Pradesh government, which will show the similar trend. Finally, these WBE results provide the baseline information for the researchers and policymakers to implement the epidemiological studies in the wastewater domain, and also these provide the early warning signals as asymptomatic people also discharge these RNA fragments. It will give the early warning signal to the selected community. And I would like to thank you to my directors, Mr. AICT, and my supervisor and organizing committee of the C conference. Thank you.
Yeah, thank you. There's only one quick question. Just
Actually, Manpati is not online, so we can move to the next presentation. That is paper number 139, Manvita. I think video is there. Wow.
For the study, we have selected Sirikuram village as the 65% of the population is involved in the dyeing practice. 1,100 people, which produces around 0.28 ml of domestic waste water. Dyeing practices are done during the early hours of the day and on a daily basis and generally produce 0.12 ml of the daily dye effluent, adding up to 0.4 ml. The wastewater is simply flowing down the drains and into a nearby surface water body and requires immediate retreatment. Samples were collected in the month of April 2021 and at the end of each line and analyzed for various physicochemical parameters. Process of the village, 3-unit processes have been adopted: physical process, physicochemical process, biological process, and if necessary, we're doing the tertiary treatment and disinfection if it's being used. So I'm currently focusing on the biological treatment process, where I'm adopting a hybrid treatment system, which uses fed in a fed batch mode and uses facultative culture. The initial parameters are as given below, and 4 different conditions have been selected: aerobic, aerobic anoxic, anoxic, and aerobic anoxic aerobic. The results show that the COD, TOC, nitrates, and color were observed to be decreasing during the entire retention time. The amines were also observed and tested, and in condition 3, it has been decreasing and increasing, and finally decreasing, which implies that amines have been formed in condition 3 under the anoxic conditions. You can also observe that the complete removal of color has been observed after the 24-hour HID, and it has been optimized. The COD removal occurred with maximum amino reactor with efficiency, and the T removal was also tested with the removal of 70% efficiency. Mines were tested, and it was observed that after provision of aeration after 12 hours, the complete removal of mines was observed in the condition C4. And similarly with the color, complete removal of color was observed in the first 12 hours of the study, and it was visually invisible. The above results indicate that the C4 can be used to further optimize and integrate it along with electrotreatment as well as advanced oxidation HRT. Treated dye wastewater can be further disinfected and can be used or discharged to the environment safely after meeting the BIS standards. I would like to thank the Department of Science and Technology for funding for this project and the
Why is this taking 12 hours to create amines? Is it
Sir?
expedient this process like
That.
I'm sorry, sir.
Hello, sir. Good afternoon, sir.
Yeah.
Recording.
Can you hear? Yeah, please. Yeah, very, very good. Briefly. Can you please elaborate, like, very briefly, why is the degradation process is long? Like 12 hours is a nominal, or your process is better? Like 12 hours, I saw amine is degrading in 12 hours. That's what your claim is. Whether that process is normal or like it's long, or how to make it shorter if you want to do continuous process treatment. Amines, like how long it will take to degrade the amines? Hello? Amines. I think she can hear. Hey, you're muted, Manita.
Can you unmute yourself? Okay, then I think there is a network issue and we can move further. On my personal behalf, I feel apology to all the phone speakers for the problem in the software. And we tried our best for the smooth running of there were some issues. So my apology for the same. And thanks for being with us. And now I will request Professor Smee to just conclude the session.
Thank you so much. Thank you so much, Professor Paneswar, and then also like to thank all the panelists for giving excellent presentation. And I know there has been some technical glitch, but in spite of that, we were able to achieve. Actually, the one presented online was really easy to understand compared to the on-site. But anyway, so I'd like to thank all the panel members for excellent presentation and staying late, Hector and also Henry for getting up early. And with this one, I think we are going to close this session. And I think we need to return those evaluation form by email, right, Paneswar?
Yes, sir. Yes, sir.
Yeah, so
Actually,
kindly.
the lectures only for our panelists. I think audience may not got all these glitches, so you don't need to worry about it. Like audience does not have these glitches. It's the panel members who are having these kind of glitches.
Yeah.
Just
Okay, thank you so much, and stay safe, and wish you all a very happy New Year, and hope to see you maybe sometime face to face. So with this one, we conclude the session. Yeah, bye bye. Take care. So,
Thanks.
Professor
Bye-bye.
Pamjit, yeah, bye bye.
Thank
Please be
you.
online.
2 minutes.
Please be online. I would like to thank Professor Paramjit Panesar and Professor Samir Khanal for moderating the session. And the speakers of the session, our panelists of the session, Professor Hector Lewis, Dr. Palanivel Senthil Kumar, Dr. P.H. Lee, Dr. S. Venkat Mohan, Dr. R. Parthasarthi, and Dr. Amitek. Now we are going to further for this session. I would like to request Professor Ashok Pandey to give the mementos to the speakers and the moderators. I request Dr. Paramjit Panesar to come forward.
I request Dr. R. Parthasarathy to come. I request Dr. S. Venkatmohan. And the virtual panelists and moderators, society will felicitate whenever you attend the conference physically. Thank you, everyone. Happy New Year. Happy
Happy New Year.
2022.
Yeah, bye bye. Happy New Year. Bye bye, Sunita. Bye bye, Ashok.